While the causes of ALS are still not fully understood, one emerging area of focus is RNA dysfunction. In ALS, abnormalities in RNA processing contribute to the loss of motor neurons. This dysfunction is central to both familial and sporadic ALS, although the mechanisms may differ. In this video, we'll explore the role of RNA dysfunction in ALS, from the genetic mutations driving these errors to how these disruptions ultimately lead to neurodegeneration. RNA processing is an essential function that occurs inside every cell, including motor neurons. In healthy cells, RNA is transcribed from DNA, processed, then used as a blueprint for making proteins. But in ALS, this process breaks down. Proteins that help regulate RNA, such as TDP-43, FUS, and ADAR2, are critical to this process. When these proteins malfunction, the RNA processing system becomes disrupted, leading to the production of dysfunctional proteins. These proteins accumulate and form toxic aggregates inside neurons, causing them to lose their function and eventually die. ALS can be classified as either sporadic or familial. The majority of ALS cases are sporadic, meaning they occur without a clear family history of the disease. In these cases, RNA dysfunction is often triggered by environmental or genetic factors that are still not completely understood. In familial ALS, however, genetic mutations play a direct role in the disease. One of the most well-known mutations occurs in the C9orf72 gene, which is responsible for producing a protein involved in maintaining neuron function. This mutation causes a repeat expansion in the RNA, leading to the formation of a toxic RNA foci in the nucleus. These foci trap RNA binding proteins, disrupting their normal function and preventing proper RNA processing. These RNA foci are toxic, and they create a cascade effect that disrupts the delicate balance of cellular activity, contributing to motor neuron death. One of the key players in ALS is TDP-43, a protein that normally resides in the nucleus where it helps regulate RNA. However, in ALS, TDP-43 becomes mislocalized to the cytoplasm, where it aggregates into clumps. These clumps of misfolded proteins are toxic to neurons, disrupting normal cellular processes and leading to neurodegeneration. FUS, another protein involved in RNA regulation, also mislocalizes in ALS. Just like TDP-43, FUS aggregates in the cytoplasm, causing further disruption to RNA processing. Both TDP-43 and FUS are involved in a stress response mechanism inside the cell, but when these proteins are mutated or misfolded, the stress response turns from protective to destructive, leading to ALS. ADAR2, a protein that edits RNA, is also disrupted in ALS. Normally, ADAR2 regulates the function of AMPA receptors, which control the flow of signals between neurons. When ADAR2 is dysfunctional, AMPA receptors become overreactive, leading to overstimulation of neurons. This overstimulation causes a phenomenon called excitotoxicity, which contributes to motor neuron damage and death. In ALS, the misfolded proteins do not remain isolated within a single neuron. Much like prions, which are misfolded proteins that spread from cell to cell, TDP-43 and FUS spread through the brain and spinal cord, causing other neurons to misfold their proteins as well. This prion-like spread accelerates neurodegeneration, creating a self-sustaining cycle that makes ALS so difficult to treat. Both TDP-43 and FUS are also implicated in the formation of stress granules, temporary structures that form in cells under stress. While stress granules are meant to protect the cell by sequestering damaged RNA, in ALS, the stress granules become too large and insoluble. Instead of protecting the cell, they become toxic, further contributing to protein aggregation and neuron degeneration. Now, I wanted to briefly talk about two final genes, specifically UNC13A and STAthmin2, which are currently a hot area of research. Scientists are actively studying these genes and developing potential clinical therapies based on their roles in ALS. The first gene is UNC13A, which is located on our 19th chromosome, and it's important for neuronal communication by helping prepare and release neurotransmitters. Now, an RNA molecule contains regions called exons and introns. During RNA processing, the cell is supposed to cut out the introns and join the exons together at a specific spot called a splice site. Sometimes the cell mistakenly uses an incorrect spot, one that is not normally used as a splice site, during the RNA splicing process, and this is referred to as a cryptic splice. This can then lead to the inclusion of unintended sequences that should have been left out. Now, one of TDP-43's functions is that it binds to the intron sequences and prevents those parts from being added to the final version of the RNA that the cell uses to make proteins. Thus, when TDP-43 is absent, it can't properly regulate how UNC13A's RNA is processed. As a result, a part of the UNC13A gene that shouldn't normally be included gets added by mistake. This results in a cellular process that degrades the faulty RNA, causing a loss of functional UNC13A protein. Reduced UNC13A protein will lead to an impaired neurotransmission, contributing to the worsening of the ALS symptoms. Cells lacking UNC13A have showed that they release neurotransmitters at nearly a 4 times slower rate. Absence of UNC13A also decreased the release of dense core vesicles by up to 60%, which are vesicles that contain neuropeptides, neurotrophic factors, and other regulatory substances which develop neurons in the adult brain. Now, the second gene is stathmin-2, which is specifically expressed in the nervous system for developing and maintaining the axon of a neuron. While excessive or reduced levels of stathmin-2 cause the collapse of the tip of a growing neuron or suppress the extension of dendrites and the axon of a neuron, normal levels will stimulate the outgrowth of a neuron's axon and dendrites, which is vital for communicating with other cells. Now, functional TDP-43 binds directly to stathmin-2 RNA to maintain normal splicing. The pathological TDP-43 drives cryptic splicing in the stathmin-2 RNA, leading to the production of a non-functional RNA. Reduction of TDP-43 or stathmin-2 in human motor neurons inhibited the regeneration of axons after they were damaged, and notably the restoration of stathmin-2 restored the outgrowth of axons and dendrites as well as axon regeneration in the absence of TDP-43. Understanding the complex relationship between RNA dysfunction and ALS is crucial for finding effective treatments. Researchers are actively exploring potential therapies that could target the misprocessing of RNA, the aggregation of toxic proteins, and the restoration of normal RNA regulation. For example, some approaches are focused on using small molecules to break up protein aggregates, while others are investigating gene therapy techniques that could correct the RNA processing errors caused by mutations. These methods offer hope for slowing the disease and improving the quality of life for people with ALS. Though this process is slow, advances in understanding RNA dysfunction and the mechanisms behind ALS are moving the scientific community closer to viable therapies. The goal is to halt or even reverse the progression of the disease, giving patients more time with their loved ones. Though ALS remains an incurable disease, the increasing understanding of RNA dysfunction offers hope. With continued research, breakthroughs may soon be within reach. Together, we can make a difference in the fight against ALS.